afternoon, everyone. Thank you for connecting to the first liver seminar of this year. It's a pleasure for me to introduce our guest today, the Professor Pierre-Emmanuel Ratout. Professor Pierre-Emmanuel is a clinical specialist in hepatology with a PhD in vascular biology 2011. He is professor of hepatology at the University of Paris and Hospital Bijon since 2016. He is the head of the Splanchnick Hemodynamic Laboratory at Bijon Hospital since 2012. He's also leading an INSERM team dedicated to the study of the role of vessels in the liver diseases at the INSERM Unit 1149, and his research is focused on the role of the vessels in the liver diseases. Specifically, his interest in primary vascular liver diseases, Budd-Chiari syndrome, portal vein thrombosis, and intrahepatic non-cirrhotic portal hypertension, as well as in vascular involvement in common liver diseases development and complications. He's been chair in the ACIL Scientific Committee, a member of ACIL Governing Board, and recently has been appointed as chair of VALDIC, Vascular Liver Disease Group. VALDIC is conducting multi-center research projects. Those studies focus on the incident, direct causes and consequences of vascular liver diseases, and investigate mechanisms that lead the pathological changes in the liver vascular and they test new effective treatment. And finally, the lead of Ratout Lab with the main three current research directions: liver sinusoidal endothelial cells, extracellular vesicles in liver disease, and portosinusoidal vascular liver disease. So before starting, welcome and thank you very much to be here with us, Pierre-Emmanuel.
Thank you very much, Edilmar, for this very kind introduction. Thank you to you, your dear Rafa, for inviting me. It's really a great pleasure to be with you. Really a great pleasure too to see many friends and collaborators participating to this session. So I will talk today about extracellular vesicles, focusing mainly in cirrhosis. And I would like just to remind you to start with what extracellular vesicles are. There are basically three types of extracellular vesicles. The smallest one, called previously exosomes. Now it more referred to small extracellular vesicles. These ones are very small, the size of a virus, and they've got a formation process different from the largest one called microvesicles. The smallest are derived from multivesicular bodies fusing with the plasma membrane, while the largest one, called previously microvesicles, are derived directly from the plasma membrane. Even bigger are apoptotic bodies, but I will not talk too much about those ones because they are more difficult to differentiate, particularly in the plasma, from the platelets because they've got the same size. So let's focus in this talk on exosomes, also called small extracellular vesicles, and microvesicles, also called large extracellular vesicles. So here you can see an electron microscopy picture of a patient with hepatitis C. You can recognize here an hepatocyte with the nucleus and the mitochondria. Here are erythrocytes. And if you look at those debris here in the sinusoid between the erythrocyte and the hepatocyte, you can see that these debris are in fact vesicles, and vesicles having a size of large or small extracellular vesicles. If you take a look at another place in another patient, you can recognize again an hepatocyte, again an erythrocyte. Here is a Kupffer cell, and here this small cell is an endothelial cell. And if you use a higher magnification, you see that this cell, this liver sinusoidal endothelial cell, is blebbing and releasing an extracellular vesicles, and maybe is sending an information from this endothelial cells to the neighboring hepatic stellate cell. So my talk will be divided into two parts. First, I will talk about extracellular vesicles, EVs, as biomarkers, and I will refer to a review article we published recently on this topic. And in the second part, we will see how extracellular vesicles can transfer information from one cell to another. You are very familiar with all these chronic liver diseases, with the causes on the left-hand side, leading most of them to cirrhosis that can lead to liver cancer and decompensation of cirrhosis. But in the perspective of biomarker, we can easily recognize that we have many questions in liver diseases. In purple, you've got the prognosis questions, in orange, the diagnosis questions, and in blue, the diagnosis questions. And it is, this is something more for the youngest amongst you. It is important to recognize well these questions because the tools we'll use will be different if we want to diagnose alcoholic hepatitis, if we want to early diagnose HCC or predict the development of HCC or predict the compensation of cirrhosis. And vesicles in some way could be useful to answer several of those questions. To use extracellular vesicles as biomarkers, there are several steps. First, rationale. Why can we use extracellular vesicles as biomarkers? And then two types of approaches: hypothesis-driven approach, the one we used in the past, and now agnostic approach. So the rationale is the following one. Cells are releasing extracellular vesicles when they are undergoing a stress. They are releasing some vesicles in physiological conditions, but when they are undergoing a stress, they also release extracellular vesicles, and special maybe extracellular vesicles because it has been well demonstrated that the the concentration in extracellular vesicles released, but also the composition of the extracellular vesicle release depends on the cell type and on the stimulus initiating cell bleeding, meaning that extracellular vesicles released from hepatocytes will be different from extracellular vesicles released from endothelial cells or other cell types. But in cirrhosis, we know that the liver is injured, but we know also that many other organs are impaired. Immunity is, of course, impaired. The kidney, the adipose tissue, the pancreas, the gut, and other organs. And the hypothesis beyond using extracellular vesicles is that using several subpopulations of extracellular vesicles measured in the blood, we could capture the complexity of cirrhosis and reflect the outcome of the patients using several subpopulations of vesicles. But before starting measuring extracellular vesicles in the blood, we should also consider that what we are going to measure is the results of a balance or maybe an imbalance between clearance and production, like for everything. It leads me to the question of the clearance of extracellular vesicles, or we are talking about clearance, but it's in fact an uptake, because for us hepatologists, this uptake by liver cell, for instance, may not be just a clearance, maybe a way to transfer information, but we will go to that in the second part. So what do we know about clearance? There have been many studies and also systematic review, and I'm sharing here with you a recent systematic review summarizing the clearance, the uptake of first small extracellular vesicles. You can see on the left part the organs, and the uptake is when the organ is red, very high, and when the organ is green, very low. And you can see that very rapidly in the first hours, the liver takes up large proportion of small extracellular vesicles, also the lungs, or less for other organs. The spleen plays a significant role. And then it goes to the urine, as you can see here. What about large extracellular vesicles? Again, the liver plays a big role, the spleen a little bit, the lungs a little bit, and not so much the rest. So for us, hepatologists, we can speculate that an injured liver, a diseased liver, a cirrhotic liver, is associated with a decreased clearance of extracellular vesicles. Likely, it could explain some of the changes we observe in patients with cirrhosis. What about production of extracellular vesicles in the setting of liver diseases? Just an example, because it has been obtained in patients. What we did is that we measured in patients, 76 patients with cirrhosis, plasma hepatocyte-derived extracellular vesicles. And we compared what we measured of extracellular vesicles in the plasma with histological lesions observed in the liver of the patients. And this is a systematic review of the slides of the patients. And we observed that extracellular vesicles concentration was higher when there was more activity in the liver, more fibrosis, and also more apoptosis. So likely in patients with cirrhosis, there is a decreased clearance of extracellular vesicles and likely an increased production going together with inflammation occurring in the liver and apoptosis occurring within the liver. So we need to have the question in a biomarker perspective, we need to have good questions. We need to take into account the clearance, but pre-analytics, meaning how we prepare the samples, is key and really should be emphasized. First, when you want to do some measurements of extracellular vesicles, you should take care of the type of tubes. And we discussed that this morning with some of you. Citrate tube are usually recommended, and also EDTA. Then the centrifugation speed at 2000 G twice is useful to get rid of all the cells and to obtain plasma with vesicles. Here you can see the summary of several guidelines on extracellular vesicles, and fortunately they all concur with the type of anticoagulant, the using a quite large needle, remove the cuff rapidly because hemolysis, this is really a big problem. The tube should be stored, filled properly and stored vertically, and really throw away the hemolyzed tube, and double centrifugation is useful. So these are the conditions you should use if you want to analyze extracellular vesicles in the blood of the patients. So then you obtain plasma with vesicles, and here the question that comes is, what is the purpose of your analysis? Is that only discovery on a limited number of patients, or is that high throughput where you want to measure hundreds and hundreds of plasma samples? And the technique that should be used really differs in a discovery or in a high throughput perspective. I will not go into detail into the different methods to be used for that purpose, but for separating the vesicles, you can use centrifugation, gradient, immunocapture, or the one we currently prefer, which is the size exclusion chromatography. This is for discovery, but if you want to do high throughput, you can use filtration of the plasma and also use some kit to precipitate or capture the vesicles. Some words about the filtration, because this is something that I will use in the next slides to measure extracellular vesicles. The principle is quite simple. You obtain plasma without platelets, without cells, and you filter this plasma through a pore, 0.1 micrometer, and therefore you remove the large extracellular vesicles, and you can then run an ELISA on the plasma with and without large extracellular vesicles, and the difference between both represents the portion of the marker you are measuring by ELISA carried by extracellular vesicles. So it was for separating, and we started with the detection with ELISA. There are other strategies to detect. High sensitivity flow cytometry, meaning flow cytometry with dedicated devices specifically designed for extracellular vesicles and used only for that purpose. This is a good technique, but not to run hundreds of samples. And qPCR is also useful for specific purposes. We can discuss on the techniques if you want later on. So it was the hypothesis, it was the rationale. Then what can we obtain with a hypothesis-driven approach? This is what we did as an example some years ago already, where we consider that in patients with cirrhosis, there might be changes in hepatocyte, extracellular vesicles, and in vesicles derived from leukocyte, endothelial cells, lymphocyte, and erythrocyte. And we chose some markers, specific or more or less specific for some populations. And we measured that in the plasma of patients with cirrhosis and of controls. And we observed that patients with cirrhosis have higher concentration of vesicles of leukoendothelial, leukocyte, lymphocyte, erythrocyte, and hepatocyte origin. And if we take into account and focus now on vesicles of hepatocyte origin, we observe that the concentration of hepatocyte extracellular vesicles increased with severity of cirrhosis. So based on these results, it could suggest that hepatocyte extracellular vesicles could be interesting biomarker maybe in patients with cirrhosis, and we started a prospective study. It was a collaborative study between our center in Paris, in Beaujon, and the Hospital Clinique in Barcelona with the team of Juan Carlos. And on the left-hand side, you have the Beaujon cohort, and the right-hand side, the Barcelona cohort. And we observed exactly the same. Again, an increase in the concentration of extracellular vesicles of hepatocyte origin with the severity of cirrhosis reflected by the Child-Pugh score. On the left-hand side in France, on the right-hand side in Barcelona. But when we analyzed the again in Paris, on the right-hand side in Barcelona, we observed that patients with a high MELD score, meaning a MELD score above 15 and high concentration of hepatocyte extracellular vesicles, have a high rate of death at 6 months as compared to other groups. It was true in France. It was also true in Spain. So my interpretation of those data is that if you are in front of a patient, a severe patient, MELD above 15, the one who will die within 6 months, this is the one who has still an ongoing liver detrimental process reflected by high concentration of hepatocyte extracellular vesicles. So we continued on that direction, and I'm sharing here with you unpublished data that might be presented at ILC if selected. We moved from several severity stages of cirrhosis to here only compensated patients with cirrhosis from the prospective French cohort called CIRAL. And here we analyzed the cumulative incidence of decompensation in these patients with compensated cirrhosis. And we observed that patients with high hepatocyte extracellular vesicles and high fibro test have a very high rate of decompensation at 2 years as compared to other groups. You can notice that the rate of decompensation is as high as 60% in this patient population. So it was some examples of hypothesis-driven approach, but now we have new tools allowing us to use agnostic approaches. The most interesting one, this is the size exclusion chromatography. Why? Because this tool allows us, as you can see on the left-hand side, you can see the plasma of a patient with cirrhosis, very yellow, and this allows us to separate, using this column, the vesicles here from the rest of the protein of the plasma. And this is very important because if you want to do specific analysis on the extracellular vesicles, you have to be pure and not be contaminated by all this bunch of protein. So now, thanks to this technique, we are able to do that and to perform typically proteomic analysis, but others do mere microarray, for instance. So let's start with some data on NASH and NAFLD. These are the results of a study performed by the Feldstein Group, and René Bosch contributed to that work, where the authors observed that healthy individuals, in green there, have a composition of vesicles in terms of protein that is not so far from pre-cirrhotic, but is very different from cirrhotic NASH patients. At our center, we focused not here on cirrhosis, but on healthy controls and patients with NASH at the early phase, and did a principal component analysis, meaning that in this graph, one dot represents one patient, a blue dot, this is a healthy individual, yellow dot, this is a patient with steatosis, and we see that the composition of the protein in the vesicles of those patients, in the blood of those patients, is very different according to the status, steatosis in yellow and healthy in blue. And when we analyze the pathways that are the most represented in these vesicles, we see that the liver is something highly deregulated in the vesicles of patients with steatosis as compared to healthy individuals. What about cirrhosis when we use similar approaches? Here we did chemical analysis on extracellular vesicles from patients with Child-Pugh-A cirrhosis in blue, Child-Pugh-C cirrhosis alive at 6 months in pink, and Child-Pugh-C cirrhosis but deceased at 6 months in green. And again, we see that the composition of the protein differs strongly according to the status of the patient. Of course, between Child-Pugh-A and Child-Pugh-C, but interestingly, between Child-Pugh-C alive at 6 months and dead at 6 months. So this is really the basis for further studies. And if we, for instance, look at the pathways implicated between or deregulated between Child-Pugh-C deceased and alive at 6 months, we are interested to see that immunity and metabolic processes are deregulated. Now, some words about even more advanced stage, focusing on decompensation of cirrhosis. And this is part of the European project, Decision Project, which is a European project involving 21 institutions and lasting for 5.5 years. Some of you are participating to this project, and among what we are analyzing in this project to predict the outcome of patients with acute decompensation of cirrhosis, there are extracellular vesicles, and I'm going to share with you the first data we obtained in this ongoing project. We performed again the same proteomic analysis on 37 patients with acute decompensation of cirrhosis, and we observed that in green, patients with acute decompensation of cirrhosis but not developing ACLF have a different composition as the one developing ACLF or having ACLF at inclusion in terms of protein composition. Based on this approach, we selected some subpopulations of vesicles that seem to be different between the group of the patient and the outcome of the patients. And we tested those patients first in a subgroup of the PREDICT study. This European multicentric study, including more than 1,200 patients with acute decompensation. And then in the PREDICT and ACLARA study, ACLARA, this is the same study, but in South America. And here is the first result we obtained. We found that one vesicle of hepatocyte origin has a concentration that differs across the group between acute decompensation of cirrhosis and healthy. This is very obvious, but also according to the outcome of the patient not developing a new acute decompensation have lower concentrations than the one developing in the next weeks an ACLF. So if we look back and look also to the future, what is quite interesting in the development that we did and presented you is that there is a close link between the technical development and the progresses that we are able to make and the number of patients we are able to analyze. Initially, 10 years ago, we used flow cytometry, which was good and provided us with some first result, but was quite low. And when moving to the filtration ELISA, we were able to measure more patients in a more reproducible manner. And we were able then to show that hepatocyte extracellular vesicles can predict the death of the patients. Now, thanks to proteomic analysis, we open our mind and find new candidate biomarkers. And these candidate biomarkers can be validated in large cohort of patients with cirrhosis. What is the future is going even faster, being even more reproducible, and testing even larger cohort of patients to validate, to see, and we'll discuss that later on, what are the variables that could influence this concentration in the general population. And here, this is quite funny. If you take a look at the process of the development of a biomarker, this is a quite long process. It goes from preclinical assay development, clinical development, and assay implementation. And you can see here that according to the disease type in our field, alcohol, hepatitis C, hepatitis B, cirrhosis, HCC, the step where we are varies a little bit, but you can see that cirrhosis, we are quite far already with prospective studies. Now, in the second and last part, I will give you some examples of the role of extracellular vesicles as vector of information from one cell to another. And we summarize those data in a review article that we published with the group of Vijay Shah recently. All of you are very aware of cirrhosis involving first the liver, of course, and thereafter changes occur in the arteries and in the portal vein. So what is the role of extracellular vesicles in the crosstalk within the liver? Let's focus on this part in brown. Here is a summary of the involvement of extracellular vesicles in the crosstalk between cells. You recognize an injured hepatocyte, you recognize hepatic stellate cells, quiescent, activated. Here again, an activated hepatic stellate cell and a cholangiocyte, and here is an endothelial cell. So everybody is there, maybe some macrophages are missing. Hepatocyte, and this is data obtained by the Feldstein group. Hepatocyte can release extracellular vesicles that can act on quiescent hepatic stellate cells and transform these quiescent hepatic stellate cells into activated hepatic stellate cells. This is through the miR-128 carried by extracellular vesicles. The same group, the Feldstein group, also demonstrated in another paper that also endothelial cells can release extracellular vesicles and again activate hepatic stellate cells. But in turn, other group demonstrated that activated hepatic stellate cells can activate quiescent hepatic stellate cells, and this induces a vicious circle and favors progression of fibrosis. On top of that, and you can see that it leads to fibrosis deposition, all these processes driven initially by hepatocyte and by endothelial cells. The other process is angiogenesis, and in fact, several cell types can signal to endothelial cells to favor migration and tube formation. It is true for hepatocyte that can activate endothelial cells through vanine, but it is also the case for hedgehog carrying extracellular vesicles derived from hepatic stellate cells. This is work from the Videsha group and from cholangiocyte, paper from Chantal Hussein in Paris. And therefore, you can see that these extracellular vesicles carry information from one cell to another, but it is quite clear that in cirrhosis, they contribute to fibrosis deposition and to angiogenesis. If we now go back to the liver and consider the progression of cirrhosis, we know that the secondary event, this is the vasodilation, and also that activation of coagulation could contribute to cirrhosis progression. Some years ago, we demonstrated that in arteries, extracellular vesicles present in the blood of patients with advanced cirrhosis, Child-Pugh B and C cirrhosis, not Child-Pugh A cirrhosis. These vesicles are able to fuse with endothelial cells of arteries and provide to the COX pathway some substrate that leads to the release of prostacyclin, and it leads to vasodilation of the artery and thus favors portal hypertension. Also, we observed in other studies that the blood of the patient with advanced cirrhosis, Child-Pugh C cirrhosis, carries extracellular vesicles expressing tissue factor, and tissue factor, this is the primary initiator of the coagulation cascade, meaning this is a highly procoagulant molecule, and it might contribute, and this is what we observed in mice, to activation of coagulation in those patients. And some of you recently demonstrated that coagulation activation likely contributes to progression of cirrhosis. So here you've got the whole picture. Vesicles contribute to intrahepatic changes associated with cirrhosis, but also to extrahepatic changes associated with cirrhosis, meaning portal hypertension and the procoagulant state. With that, I would like to conclude by saying that circulating levels of several subpopulations of extracellular vesicles are increased in the blood of patients with cirrhosis. Extracellular vesicles might reflect ongoing liver injury and thereby predict the outcome of the patients. Pre-analytics, this is really key. And now we have many techniques. The problem is more which one to use, but maybe we will discuss that together later. And putting together some subpopulation of extracellular vesicles might be a useful tool to better diagnose patients with liver diseases and better predict the outcome of patients with liver diseases. In terms of vector of information, this is quite clear that vesicles contribute to the transfer of information from one cell to another within the liver, but also act remotely to arteries by inducing vasodilation, and also likely contribute to activation of coagulation of patients with cirrhosis. I would like to thank my clinical department on the left-hand side and my lab dedicated to the vessels and liver diseases. And here are the funding agency, and I would like to thank you for your attention. So this is a very important question. For all those markers, we know the protein, of course, and we look at the known cell of origin. So some are specific or quite specific. Others are less specific. The one I showed you is quite specific for the hepatocyte. Of course, basically no protein is specific for nothing, and usually the more specific you think it is, the less known is the protein, meaning that when knowledge increases, then you realize that it's not so specific, and it is it alludes to the work we have in front of us, meaning after having found an interesting signal in the population, it's interesting to know the variables that impact those levels in a healthy population. But yeah, this is an high level of specificity, but not 100% for sure.
Okay.
Yeah, this is a very interesting question. So this is the occasion to discuss a little bit the setting. If you're in the lab and you want to run some samples, 10 sample or whatever, you can do what you're proposing. This is a nice option to capture vesicles using some markers known to be associated with vesicles. There is some discussion about the one you are mentioning because they are more for small extracellular vesicles, maybe less for large extracellular vesicles, but it is a way to proceed. If you want to go to high throughput, then it's another story. So to answer you more clearly, what we do is the filtration system I showed you, because we use that many times, publish that. This is a highly reproducible and sensitive technique. I'm not saying this is the answer to all the problems, because it works for hepatocyte extracellular vesicles, but it will likely not work in other settings. But this is a good option to try. And to allude to discussion we had this morning with some of you,
yes,
there is not a unique, fantastic technique. It depends really on the setting and on the condition, but yeah, this filtration ELISA technique, this is one option that we like quite a lot. Yeah, this is the big question. To go a little bit more into detail, so this is kind of the holy grail, you know. Each one of us is interested in some cells. Some of them are quite rare, or even not so rare. Take just the example of liver sinusoidal endothelial cells. They are not so rare, but the problem is that the markers they are harboring are not so specific. We would be very interested to find some markers, but then imagine you go to the whole blood, where there are tons of vesicles released by platelets, because this is the major population, but also red blood cells, etc., and all the tissues. So the first challenge is you will need to find a few vesicles out of a large number, and then this is the specificity of the cell type. This the specificity of the marker depending on the cell type. Meaning, if you want liver macrophage, you need a marker that is specific for liver macrophages and not expressed in other places in the body. And to add another level of complexity, take into account the vascular beds, because if you take the blood from the arm of the patient, it means that this blood went out of the liver, because usually we are interested in the liver, out of the liver, went through the lungs, and you saw that the lungs is a place where a lot of vesicles are cleared, and then goes to the arteries and the arm of the patient. So there are several difficulties, and another one, but in this setting, to give more positive view, I think that the technique to use is something like more flow cytometry, because flow cytometry, it's not so good, I think, to run a 1000 plasma sample. But if you want to find a population that is quite rare, then I think this is a useful technique. So to answer in few words,
yeah,
it's not easy for sure, but it's possibly feasible depending on the cell of origin, and flow cytometry would be likely the technique I would recommend. So regarding basically large EVs, they are derived from the plasma membrane of the cells they are derived from, meaning they take part of the plasma membrane of the cell they are derived from, and they also take with them a portion of the cytoplasm and even some organelles, because it has been shown that mitochondria or part of mitochondria can be within vesicles, meaning that the composition includes all what you mentioned. It has been shown, for instance, for cytokine that some of them can be carried clearly by extracellular vesicles. So it's everything is kind of possible within these vesicles: lipids, protein, and also nucleic acids. And consider also in terms of pathophysiology that this release is not random, because small extracellular vesicles have a specific way of release via multivesicular bodies, and large extracellular vesicles another. And it has been shown that the content not only depends on the cell type, not only on the stimulus, but also on the type of vesicles, and you have a different content in small as compared to large extracellular vesicles.
Okay. I think so.
Yeah, this is a super important question. This is this is for us the next step. Clearly, I completely agree. If we want to the first step was to show that this is potentially interesting, and then put firmly this is interesting. But then comes your question you are leading. If we take a single patient in a stable condition 5 times in a row, do I have the same numbers or not? And what are the variables that could influence? So this is things that should be addressed now. But so far it's unresolved questions. Very important, but unresolved questions.
Okay.
So yeah, with the amount, not the total amount, but the amount in terms of cargo, yes, in terms of specificity, some subpopulation of vesicles clearly increase with progression of cirrhosis and can predict the outcome of the patient. This is true for hepatocyte-derived, some subpopulation of hepatocyte-derived extracellular vesicles, but not only. So yeah, clearly.
Okay.
I don't know. No, this is a good question. The question is, what is the part related to
contribution reduce the clearance due to hepatic insufficiency? Yeah.
What is the part related to the clearance? What is the part related to the production? This is an interesting question, but we don't have the answer so far. Yeah, it would need to. It could be addressed, by taking patients where we cure the cause, hepatitis C, very severe patients. So, but so far not been done. This is a good question. Maybe a good plan for study. Yeah, very
interesting, and maybe some reversion will be the future too. How to identify those patients with reversion and better liver function, and what happened with the EVs in the plasma, no?
Okay, so this is an hypothesis is due to the type of uptake, because the uptake, we are looking at saying this is the lung, this is the liver, this is kind of very gross or approximative view, because in each of these organs, there are of course many cell types, and the cell types contributing vary also between organs. For instance, macrophages of course contribute, but endothelial cells also contribute, and the molecules involved in the uptake vary at the surface, for instance, of endothelial cells, meaning that the type of vesicles that will be taken up will not be the same according to the organ, according to the cell type. So with that, you see that there is some specificity in terms of clearance for the cell and the organ, meaning that different vesicles will have different clearance. And from that, you understand that the clearance of the small will be different from the clearance of the large one. I don't know if I've been clear. This is a heavy topic. The clearance or uptake is quite heavy topic. But for us, I think we should prefer uptake rather than clearance, because I don't think this is just the liver is not just a trash can. This is an organ that takes up and does a lot of things with what it takes up. So this is
No?
this is a very important point that I did not address. This is using extracellular vesicles as therapeutic target for sure, but also therapeutic agents, meaning that there is a lot of work on mesenchymal cells and as a treatment, but moreover, there is a lot of work on mesenchymal cells derived extracellular vesicles, because in terms of rules, it is easier to manage extracellular vesicles than cells, and some companies are creating, and in France there are several, they are producing a lot of vesicles as therapeutic agents to treat locally or even to inject to patients. And even more, there is very interesting approaches where you can load a drug within a vesicle, and this is typical for chemotherapy, for instance, where it would be toxic, but this vesicle you load with a drug, and you can target that to a specific place where it will act, and you decrease the toxicity of the systemic toxicity of the drug. So on top of the biomarker approach, on top of the vector of information, there is this possibility of using vesicles as therapeutic agents. And to answer a little bit better the questions that was asked by Paula, in terms of therapeutic target, it is difficult because the processes leading to vesicle generation are quite ubiquitous, meaning that there is not so far a pathway to target that would suppress some vesicles without side effects. So what we usually do, we target the process responsible for the release of vesicles, but it is of course less specific. So personally, I did not do RNA so much, but close collaborators did. I don't think there is a best technique. You can do RNA sequencing.
Total or microRNA?
No, total. I don't think that there is a better approach than the other.
Okay, but are you agree that if we are gonna do the RNA-seq, the best technique to concentrate to isolate the EVs is SEQ?
Yeah.
Chromatography more than ultracentrifugation.
I think so. But SEQ, as we discussed this morning, size exclusion chromatography comes first, and thereafter you do a centrifugation. But this is size exclusion chromatography gives you a very clean population of vesicles. So now we really many of us tend to use size exclusion chromatography as the first step in this kind of experiment. The problem with size exclusion chromatography, this is very labor intensive. So if this is to run 20 samples or 30 samples, this is fine. If this is to run 500 samples, this is not possible.
Yeah.
This has been analyzed by several studies with very conflicting data. So there is no firm conclusion on that. I'm not sure, really not sure. The problem of vesicles derived from platelets is that the good thing is that they are very, very abundant, but the problem is that they can be activated very easily in an artifactual manner. So you never really know if this is something that is really relevant or not. And very large studies have tested that in other fields with negative results. So I would not put too much money on platelet-derived extracellular vesicles. What we know, something interesting is that CRP can be carried by extracellular vesicles.
Okay.
And this is a paper not in the liver field, but published in Nature Communication. And the interesting thing is that this CRP carried by extracellular vesicles is in a more active form than the free one, because it is a pentamer, and the fact that it is on vesicles changes the activity. So with regard to what you said and the really important role of CRP as a biomarker in chronic in cirrhosis, basically, it is attractive. I don't have the answer. I know this is something that to me seemed really to be tested, but we never tested it so far. So something, maybe something to keep in mind, I think. So yeah, this is agree with that. It was just the first result that I wanted to share with you. We are in the process of analyzing all those data. So for sure there is an overlap between pre-ACLF and AD patients. Still a significant difference. So the goal now is to see if we combine some vesicles, if we try to separate better these two groups, but I completely recognize what you are saying. It was just an example from the very early steps of proteomic analysis to large cohort of patients, but the analysis is not finished at all. It is just starting. And the role of EV in clinical practice, the first one I would see, I think, is the biomarker role, to better refine the prediction, and maybe more in patients with compensated cirrhosis and in decompensated cirrhosis, but with the data I showed you from the CIRAL cohort, but again, it needs validation, etc. So it's not for tomorrow, I think, but over the years, progresses are being made, I think, and the field is clearly expanding incredibly. 10 years ago, I was thinking that I don't know what this field, maybe this field will disappear, and in fact, it just exploded completely and expanding a lot. So, which is good because many progress are being made. So, but I think that the next step is biomarker results and vector of information.
Perfect. So there are a lot of to study in this field, I think, for all of us. And thank you very much, Pierre-Emmanuel, because I think that has been a very, very interesting talk. A lot of questions have arrived, have been a very nice discussion. And thank you very much to be with us today. And well,
thank you very much. It was really a great pleasure to be with you. Thank you for all these questions. And I would like to thank you, to wish you a good afternoon.
You too. Thank you very much. Thank you, everybody, to be here with us, and see you soon.
Bye.
Bye-bye.